This short clip will cover examining data integrity and texture using the zonationTool macro. The Miller glia cells present a stereotypical apical to basal structural pattern, as we can see here with the regions labeled from 1 to 5. Looking at the entire fish retina, region 5 would be located here, whereas the apical region 1 would be here. Our input is a 3D image composed of several slices of two-dimensional images stacked together. The MIP is the maximum intensity projection. This selects pixels of the highest intensity from every slice to construct a 2D image. We then take this 2D image and rotate it by 90 degrees to obtain the representation we see here. By collecting the maximum intensity projections along the x-axis of this representation, we can produce a one-dimensional vector consisting of only the y-axis. The result of this process will be the conversion of a 3D image into an apical basal intensity vector. Plotting of these results can allow us to understand the relative positions of subdomains of the cell and compare the intensities at different stages of development, as we can see on graph G and H, both representing the same data in different forms. We can see that from 24 hours post-fertilization to 96 hours post-fertilization, the subdomains' intensities and their positions have therefore changed. We go to the macros folder, drag and drop the zonation tool, and we click on Run. We are here presented with a table that asks if we'd like to perform intensity plotting, and here we confirm with a yes. The second option is that of scale normalization. We suggest that you run both with and without normalization to check with your data and see if either of them works better. What this does is that it stretches the images to similar sizes, but for images that contain significant differences, it may not make sense to stretch them, as it would lead to distortion. We confirm our choices by clicking OK, and we will run the macro on the 2C-dir input folder. 2C-dir was the output of the subregion tool, hence a 3D stack that has been normalized using that macro. Once the macro has completed running, you can check the output folder located on the 2C-dir. This will show a document titled zonationTool Profiles and zonationTool Results. If these zonation profiles are then plotted, we can obtain a saliency map that can visually represent the different intensities. Thank you for watching, and we wish you the best of luck with the Gliomorph tool.